Single here. Okay, so last time I recorded, I made you a video called Introduction to Genetics, and it was basically going over some introductory terms to understanding very basic genetics concepts, okay? So last time we were introducing things like genotype and phenotype, and what it means to be dominant, what it means to be recessive. And so today we're going to continue that practice by introducing monohybrid crosses and Punnett squares. So this is where you can really see a lot of things take shape. So let's just jump in and start practicing and see how it goes. Okay, little refresher. If something is a dominant trait, it's probably going to be represented by a capital letter. Okay, usually the problem will give you the letter that it wants you to use, but not always, but just as long as you understand dominant traits are capitalized and recessive are lowercase, then that'll take you a long way. Okay, so I've put some sample problems in front of us. I thought we would work through them together, and that way we can apply the vocabulary we learned last time and learn some new words this time. First and foremost, what is a monohybrid cross? So up here, it says complete the following monohybrid crosses. A monohybrid cross, so mono means one, okay? And then a cross just means when, like when something says cross this with this, it actually just means if you take this and breed it with this, what will the offspring, like the likelihood of certain traits be? So it's really just you're pretending that two genes are being bred or crossed in that way. So monohybrid, mono meaning one, just means we're only going to focus on one trait at a time right now. So that's why you only see the letter G in front of you. So in this case, I believe we're coding for pea plant color. Yes. Okay, so the color of the peas. Yeah. So they want us to use the letter G in that example. And they went ahead and worked one for you. So I thought that was really nice to see. So let's go down to where it says number 1 and let's do that one together. All right. Let me shrink myself. Scoop. And it says in the description, complete the following monohybrid crosses, draw a Punnett square, list the ratio, and describe the offspring. Be sure to remember that the capital letter is dominant. And then it gives you an example of a green pea plant being crossed with another green pea plant. Okay, so number 1, a green pea plant, big G, little g, so that's heterozygous. Remember, hetero actually means opposite or different than. And so heterozygous just means there's one big letter and one little letter. Is crossed with a yellow pea plant, little g, also known as homozygous recessive. Homozygous means that both of the letters are the same, meaning they're either both big or both little. And then the recessive part lets you know that they're both small. Okay, so homozygous, the same, recessive, small. So 2 small letters. All right, so let's jump in. It does not matter which parent you put up here and which parent you put to the side. It just doesn't matter as long as you understand what you're looking at and you're consistent, you're good to go. So I'm just going to pick the first parent and put the capital G right there and the lowercase g there. I'm going to color code for you so that it's easier for you to see where my letters are coming from, where my genes are coming from, but you certainly don't have to if you're practicing at home. Okay, and then parent 2 is homozygous recessive. And we've done the first part, which is just making sure that we put the parents' genotypes in the correct location. We always use the top and the left side. I don't know why people don't use the right side. We just always go from left to right when writing in English. And so I think that just sort of sticks, right? A lot of people operate from top down and left to right. So this is the same. All right, so I'm going to go ahead and fill these in. I'm going to zoom in so you can better see what I'm doing. All right, so the only rules that really apply are that capital letters have to be written first. And so right here, we go in and we drag that capital G from here And we do what's called a drag and drop. So we go boop and drag there and drop it, and then drag and drop. We distribute that G to both of those squares it can be deposited in. Sometimes you do this in math if something is in like parentheses and then there's like a number outside of the parentheses. You'll distribute that number to anything inside the parentheses. This is no different. You're just distributing that G to the boxes below it. So it kind of works the same as parentheses. All right, so I look around. Are there any other capital letters I need to focus on? No, there are not. So now we just finish up that parent. All right, and then I swap colors because I want to show you the difference. And we go to the second parent. So we distribute that lowercase g right there across this time. We go boop, boop, across. And we go down to the second lowercase g, and we distribute it across as well. All right, so we're all finished. And now you can see that each of the potential offspring will have half of their genes from mom and half of their genes from dad. So that's how you know you've done it correctly. That's part of the reason that color coding is very helpful. It lets me know that I've accounted for both parents in each box. Okay, so let's get back to the problem. So we did the cross, and then it asked us for the ratio of genotypes and the ratio of phenotypes. So looking up at the example that it gave us, it wants to know how many of each genotype existed in the squares only, and then how many of each phenotype existed in the squares. All right, so here I go. Let's pick a new color so it's not confusing. All right, so in the offspring, okay, so I'm going to go ahead and write that so that you guys know what I'm talking about, but you certainly don't have to write it on your paper. All right, offspring. For the genotypes, you list them out. So the possible genotypes that exist are homozygous dominant, which is big G, big G. Now we go back to our squares. How many of our squares have two capital G's in them touching together? 0. So we go back over here and we just write the number 0. All right, we do the next one. We do heterozygous. All right, how many of our 4 squares have one big letter and one little letter? 2. So we just write a number 2 right here. And then we do homozygous recessive the same way. How many of our 4 have 2 little g's touching? And that would be 2. And you only have 4 boxes, so that's a really easy way to check. 2 plus 2 is 4, so this one should be 0. All right, so there's the genotype. So I'm going to go over here. It'll let me. And I'm going to write genotype. There we go, genotypes. And that lets my teacher know what I've listed there. Now, likewise, it asks for phenotypes. So I'm going to pick another new color so it doesn't get confusing. I'll pick red, I think. Yeah, we'll do red. And it asks for phenotypes. Remember, phenotypes are physical appearances. Okay, so the phenotype. That's how the thing looks. All right, so usually there's only 2 phenotypes for most of these cases we're going to be talking about today. So in this case, your options are green or yellow. So you just write that down. So you say green. All right, how many of these will be green? Well, it looks like green is dominant to yellow. So anything with a capital letter will be green. So 1, 2. So 50% or 2 out of 4. So I'm going to do these as a percentage. You're welcome to do 2 for 4 or reduce that fraction to 1 out of 2. I like percentages. It just is how my brain works. All right, and then our other option was a yellow pea. All right, and those are homozygous recessive. So how many of our offspring are homozygous recessive? Looks like 2. So that's another 50%. And once again, same thing. You can do 2 for 4 or 2 out of 4 or 1 out of 2 if you want to reduce the fraction, or you can use percentages like me. Okay, so that's the answer to our first Punnett square problem. Hopefully that was really helpful. I'm going to go ahead and be quiet for a minute and just work the other one without talking through it. If you want to practice time. Okay, and just like that, we're all finished. I hope yours went well. I really enjoyed teaching you today. Next time I see you, we're going to do some more advanced things and introduce some other types of genetics, which I like to refer to as non-Mendelian genetics, and that might be new to a lot of you. Okay, I can't wait to get into some other types of problems next time. See you soon. Bye.